This video is for the Keystone Biology Review in Pennsylvania, and it's Anchor 7, The Theory of Evolution. These anchors focus on the mechanisms of evolution and the sources for evidence, as well as applying scientific thinking. This last anchor, we spent a lot of time in video 1, and so we'll just focus on some of the content for the mechanisms of evolution. Like many topics here, vocabulary is important. So take a moment, pause, and see if you can match the vocabulary terms with the definitions. Hopefully you wound up with some answers that look something like that. When it comes to the mechanisms of evolution, and we're talking about macroevolution, large changes from one species to another, we can remember some of those common evidences with this acronym FAME, F-A-M-E, fossils, anatomy, molecules like DNA, and embryology. Fossils form as living organisms are rapidly buried, put under a lot of pressure, and then decompose. As the organism decomposes, the imprint is left in the rock layer. Generally speaking, older fossils are in older rocks, lower rock layers, and newer fossils are in newer rocks. Based on some of this geology, evolutionists conclude that the older rock layers have the older species, and we can trace evolution through those rock layers. There are some questions about that interpretation, especially because of all of the species that appear during the Cambrian explosion, a rock layer that has a significant number of new fossils, but there aren't any precursor fossils underneath. Anatomy refers to anatomical structures, like analogous structures or homologous structures. Analogous structures are structures that have the same basic function. For example, fins and wings in those three animals serve the same basic function. Homologous structure means there's a similar structure in very different organisms, like the foreleg of the human, cat, whale, bat. They share a similar bone structure. That bone structure is common even though those organisms are very different. Another example of an anatomical structure that's sometimes used to support evolutionary thinking is vestigial structures. That is, structures that don't seem to have an apparent purpose. For example, hip bones in whales are thought to have been part of hind legs in whales and now no longer serve that function. The third category, molecules like DNA. In this case, we're comparing similar molecules. For example, living things make proteins. Living things make and use carbohydrates. Living things share similar DNA. So what typically happens is a gene between different organisms is compared, and the DNA of that gene is compared. And the chart is used to show the number of similarities in base pairs in a single gene between those organisms. The thinking is the more similar the DNA, the more similar or the more related the organism. So yeast are more simplistic, and they are not nearly as related to humans as pandas would be, because pandas share a larger sequence of the DNA together. And generally, that's how the chart is interpreted. From that interpretation, evolutionists can build a phylogenetic tree, a tree that shows relationships between organisms. The organisms that are least similar are farther apart on the tree. The organisms that are most similar are closer on the tree. The organisms that share the most DNA sequences are further down on the tree, and the organisms that share the most divergent sequences are higher up on the tree. The final of the four is embryology. In this case, the embryologist is studying how organisms develop in the mother's womb. And as that development is occurring, they're comparing it with how other organisms develop and using those similarities to trace evolutionary similarities. Shifting to microevolution, how species change. So not from one species to another, but a species itself and how it gradually changes based on adaptations. There are several forces that help these species make changes. Natural selection, gene flow, genetic drift, isolation, and mutation. Natural selection was Darwin's big idea. The idea that there's variety in a population because of some kind of selection pressure. Something changes. Certain individuals have adapted to survive and reproduce. Other individuals have not. Those that survive and reproduce are considered biologically fit. They pass on their genes to the next generation. As they pass on those genes to the next generation, the population takes on those characteristics. And more of the population takes on those characteristics until the population has changed dramatically. In this case, the black colored frogs are able to blend into their environment. And as a result, the white colored frogs are getting eaten. That's the selection pressure. As they get eaten, their genes are removed from the gene pool, and the black colored frogs are reproducing more to the point where the dark colored frogs have taken over the entire population. That change can be graphed in several different ways. In diagram 1 and 4, we're seeing a directional change. That is, the change is moving in a direction from the light colored frogs to the dark colored frogs. In diagram 2, you see a stabilizing kind of change, where it's the medium colored frogs that are surviving to reproduce. In diagram number 3, it's either the light or the dark frogs that are surviving to reproduce. The medium color frogs are not, and that is disruptive or diversifying selection. Sometimes migration can introduce new genes into a population. Here's an example of a population of brown beetles, and one of the beetles is transferred, somehow migrates over to the green population, and now those genes have entered that gene pool, changing that green population. Genetic drift is a mechanism that results from a disaster. So think drift, disaster. For example, you have a population of frogs, but a landslide wipes out most of the population. Only the green frogs survive. They reproduce. They pass on their green genes. Founder effect is when one or a few individuals from a population start a new colony in a new location. As a result, that new population only resembles the founders of that population, founder effect. The other is bottleneck effect. Speciation can occur sometimes by isolation. Maybe it's behaviors. So two birds, one of the birds is behaving differently, and so that bird doesn't get to mate as often and is eliminated from the gene pool. Or reproductive isolation. Maybe it has to do with species that have diverged enough that they no longer reproduce anymore. Or perhaps geographic isolation, which is what you see a picture of here, where a new river has separated these beetle populations and the populations gradually become different over time. Mutation is another mechanism that often works. In this case, chromosomal mutations can introduce new genetic variation to the population. Or it may be that DNA mutations introduce new variables into the population. I hope that helps you understand a little bit more about evolution and the processes of evolution.